Welcome to the afternoon reunion sessions. My name is Rich McNamara, and I am a member of the 50th Reunion Planning Committee. I was a double E in 67. This afternoon we will be learning more about Professor Paige Novak's research, and I do have a personal connection with Paige, as she was advisor for our youngest son Patrick when he received his PhD here at the U of M in 2012, and she did a great job. And really helped him. There we go. Paige Novak is a professor in the Joseph T. and Rose S. Ling Chair in Environmental Engineering in the Department of Civil, Environmental, and Geo Engineering at the U of M here. She also co-directs Minnesota's Discovery, Research, and Innovation Economy Initiative, advancing industry, conserving our environment. Novak's area of interest is a biological transformation of hazardous substances in sediment, groundwater, wastewater, and particularly how engineers can influence this process. She received her BS in Chem Engineering from the University of Virginia and her MS and PhD in Environmental Engineering from the University of Iowa. She was an Aldo Leopold Leadership Program Fellow in 2009 and has been the recipient of several awards, including the Sarah Evans Woman Scholar Leader Award, the Bill Boyle Educator of the Year Award, the Samuel Arnold Greeley Award, and the Paul L. Bush Award. She was named a Fellow of the Water Environment Federation in 2016, and since 2013, Paige has served on the National Research Council Standing Committee on Chemical Demilitarization. Please help me welcome Paige.
Thank you. Well, thank you all for coming. Hopefully, I'll make it worth your while missing this gorgeous day out there. I'll be talking about my research on environmental estrogens. This is work that I've been doing for a number of years. And first, I'll tell you about the problem, and it'll be depressing. And then, hopefully, I'll give you some ideas of how we're trying to solve this problem and how we're looking a little bit more long-term, thinking about how we can solve it while also solving other problems. So my area, as Rich said, is biological processes, just to kind of orient you a little bit. So I look at what the different fate of pollutants is in different engineered and natural systems based on biological processes. So what do bacteria do? And I also think about, because I'm an engineer, how can we exploit the basic needs of bacteria in order to better remove pollutants. And so the way bacteria work is they have an electron donor, which is food. For us, it's cookies and spaghetti and things. And then they have an electron acceptor, which is what they breathe, and we breathe oxygen. And by dumping electrons from their food onto their acceptor, they get energy for growth, and they produce products. And so things that I look at would be pollutants as the food or electron donor. Sometimes I look at pollutants that are the electron acceptor. And so I try to get these bacteria to do a better job of eating or breathing what we want them to. And what I'll talk about today is this idea of how we can exploit these basic needs and looking at wastewater contaminants as our pollutant, and specifically environmental estrogens. And in this case, these bacteria use oxygen as their electron acceptor, just like we do. So what are environmental estrogens? A lot of us have heard of estrogens. Estrogens are hormones. They're basically just chemical signals that go throughout the bloodstream of different organisms, and they initiate some kind of physiochemical response. And they're crucial for reproductive function and for development. And so the way they work is you would have some kind of natural hormone, you have a secreting cell, and hormones are secreted, they bind to a receptor in some kind of target cell, and this turns on a physiochemical response, something like producing eggs in a female fish. What can happen, though, is if we have some kind of external chemical, it can do a couple of different things. One is it can bind to the receptor, and it can block the binding of the hormone. And so you don't get your response. So the female fish would not produce eggs. Alternatively, you could have a situation where you don't have the secreting cell, so a male fish. And in this case, what could happen is you have some kind of external chemical, could be an actual hormone, or it could be a hormone mimic, that could bind to the receptor, and this could turn on some kind of unintended consequence, like a male fish producing eggs, which is not something that is typically observed. So where do these kinds of compounds come from? They come from us. So they come from all the products we use, the fragrances we put on, the soaps that we wash our bodies with, medications we take. That's a big one. And so all of this stuff comes from households. It also comes from industries, different cleaning products, surfactants, and then also food processing. I'll tell you a little bit about that. That's kind of an unusual one. And all of these things go to a wastewater treatment plant where that wastewater is treated, and then it goes out into some kind of receiving body. Here, it's the Mississippi River. And so it's not something that is the wastewater treatment plant's problem, although that's where we see the problem happening. So I'll tell you a little bit about how a wastewater treatment plant works so that you can kind of get a better idea about where I like to focus my work and where I might want to try to meddle and improve things. So the wastewater comes in and it goes to a system called primary treatment. And during primary treatment, we just let gravity settle out solid material that comes into the wastewater. And then that water goes on to secondary treatment. And during secondary treatment, we have bacteria that degrade and consume that stuff. They grow and they multiply, and then we settle them out, and then clean water comes out. And we've been doing this for over 100 years. We're very good at this, and we do a really good job treating normal wastewater constituents and getting, I think, very clean water out the end of the plant. But the issue is that this kind of system wasn't designed for the removal of estrogens and pharmaceuticals and some of these other trace contaminants. And the thing to think about here, too, is that if you think about all the different things that the bacteria are eating, and you start to think about the different concentrations of things, sometimes the concentrations of the pollutants that we're interested in, these estrogens or pharmaceuticals, might be really low. And so it would be like me having a whole table full of pizzas here and one tiny green bean. And I really want everyone to eat that green bean, but probably the pizza's going to go first. And so it's that kind of idea. So the question is, we know they're coming in, but are they detectable? Is this something we should even care about? And it is. These estrogenic compounds and other wastewater contaminants have been found all over the globe. They've been found in surface waters in Asia, in the U.S., in South America, in Europe, pretty much everywhere we look. And it's fragrances, it's compounds in medications, DEET. I'm responsible for some of that in the summertime. All of these compounds end up in the surface water. And they actually do have some consequences as well. I'm just going to read this because there are a lot of words here. But triclosan, this is a compound that's in antimicrobial soaps and toothpaste. It's actually been banned in Minnesota now. It's been found to alter tadpole development at relevant concentrations to what we see in the environment. And also some other work that was done at Minnesota by my colleague Bill Arnold found that triclosan will actually react with sunlight and it'll form a dioxin compound, and that'll build up in the environment. We've also seen estrogens and wastewater treatment plant effluent changing the behavior of minnows. So minnows will guard their nests, and they'll stop doing that. They're not going to be a very good protector of the nest. They won't find a mate very effectively when they're exposed to this stuff, and then they also then have trouble reproducing. And it's also been observed all around the globe that there's been a lot of fish feminization downstream of wastewater treatment plants, and this phenomenon that we call intersex fish, which this is from a paper by some researchers at the U.S. Geological Survey, which have done a lot of this work. And what they were able to see is that there's both egg tissue and sperm tissue in the gonads of a single fish. Prozac and its metabolite have been found to induce spawning in freshwater mussels. And so this can cause the mussels to spawn when they shouldn't. And it won't be very good for reproductive efficacy. And then finally, there was a really amazing experiment that was done in a full-scale lake. Up in Canada, they have an area they call the Experimental Lakes Area. And they actually dosed a full-scale lake with low levels of the active compound in birth control pills, ethinyl estradiol. And it was at a little bit higher level than you typically see, about 5 times the level you typically detect. But they saw near extinction of the fathead minnow population in the lake. And so this does kind of indicate that, yeah, we see it on an individual fish basis, but it also could have population-level, large-scale impacts. So these compounds are there, and they do seem to be having an effect in the environment. And the really interesting part of this is that this happens at part per trillion and part per billion concentrations. And so what that is, a part per trillion, that would be if we have a football field, 18 feet high, filled with sand. 1 sand grain in that entire pile would be a part per trillion. It's amazing. $15 out of the entire U.S. GDP for 1 year. That's a part per trillion. Or 1 second in 30,000 years. That's a part per trillion. And that's what we're trying to do. We're trying to get that 1 part per trillion green bean eaten. So it's a pretty tall order. And this is what we're trying to do in order to protect the environment. So what can we do? Well, from the science or engineering perspective, we can understand where these compounds come from, because that might give us a clue as to where to go after them. We can understand their fate or degradation in both the environment and in engineered systems. And once we understand this, we can hopefully use that understanding to improve treatment. We could also take a policy or economic perspective. We could try to reduce use. The big question there is how. We could ban compounds. We've done that here in Minnesota with triclosan, but a lot of times this isn't possible or isn't wise. A lot of these compounds are needed, or they're naturally excreted by women, and they're going to be there. We could introduce legislation to limit use or impact to enhance treatment, and this enhancing treatment's been talked about in Washington State. The price tag associated with this is incredibly high, and we don't even know what to monitor. And so I would say that this is probably not a wise direction to go in either. So I've focused on this top part, and I'll tell you the punchline of my talk, so then you can phase out until the end. It's really complicated. And I'll talk you through what we found here, and we think we have some really good solutions and a really good direction, but it's really complicated. But on the other side, if you go with the policy or economic perspective, I think it's even more complicated. And so from my perspective, if we can focus on what we can understand and what we can engineer, and where we're starting to think is how can we get collateral benefits, then I think we can actually make some progress. So with my research, I've worked on all of these questions, and I'll just kind of give you a highlight of some of the different things I've found with respect to understanding where they come from, what are the big problems in terms of chemicals or sources, and then looking at trying to understand their fate in the environment or in engineered systems primarily. So I'll start with the first one. So some of the things that we've been interested in are trying to figure out which compounds are responsible for most of the activity coming out of a wastewater treatment plant. So it may not be the compound in the highest concentration, but what we want to know is what's the compound that is most estrogenic? What's going to cause the most impact in terms of biota? And so that's something we've looked at. And then also been trying to figure out, are there other compounds we need to worry about in specific cases? And the reason we got thinking about this second question was that plant-based estrogens in particular exist, and they've been not, there hasn't been a lot of focus on them, I would say. These are called phytoestrogens, and they're in really high concentrations in soybeans in particular, but also things like peanuts. And agricultural production is increasing globally. And if you look at the U.S. in particular, we've been doing some things like increasing biodiesel production, and this is based on soybeans. And so it could be that as we increase the biodiesel capacity, places like Iowa, where some of our alumni are from, where I went to school, there are some other places where there are kind of hot spots for biodiesel production, and they may have some issues that we may want to look at a little bit more closely. So again, my wastewater treatment plant, what we did to kind of figure out what was the big problem was we took samples of the influent in the middle of the wastewater treatment plant and then of the effluent, and we analyzed for a bunch of specific compounds that we thought might be important, and then we also looked at activity. And we tried to figure out which of those compounds was most responsible for the activity if we could account for it. And I'll just jump over a whole bunch of data. What we found was that, and this is some data from one treatment plant. We took multiple seasons, and this is just our data from estrone. And estrone is a natural female hormone. We all produce it. Men produce it too, but in lesser amounts. And estrone was the one that was most important in terms of activity coming out of all the different wastewater treatment plants we looked at in the different seasons. And one thing you'll notice here is our influent is often lower than the next step in our sampling train. And the reason for that is that women excrete 3 different estrogens, and one of them actually turns into estrone. One of them is estrone, and one of them turns into estrone. And so you end up getting an increase in estrone across the wastewater treatment plant. And then I'll come back to some of these other patterns a little bit later. But we found out that this natural compound is responsible for most of the activity, so we're kind of stuck with it. This isn't something that we can educate people not to put down the drain. It's coming with us. We did some other things. We looked at a bunch of different industries and looked at their effluents. And basically, we took any industry we could get. We were kind of interested in some of the ones that we thought would be important, like the biodiesel. This is a little hard to see. We looked at an ethanol plant, a few different biodiesel plants, a soy oil plant, soy milk plant, a few dairies, 3 different dairies, soda bottling, a couple of breweries, barbecue meat. That was kind of an interesting one, corn wet millers. And we looked at 6 different of these plant-based estrogens, these phytoestrogens. And here I just show you the 2 that we found in the highest concentration. And this is a log scale, so it goes up by scales of 10, factors of 10. So we start at 10, 100, 1,000, 10,000, 100,000. So the concentrations go up quickly. The black line is where we think, based on the literature and some experiments we've done with a colleague of mine who's a fish biologist, this line is where we think fish impacts start to happen. And this line also corresponds with 1 part per billion or 1,000 parts per trillion. So 1,000 of those grains of sand. And what you can see is that some of these industries are really high, and some of them are weird, like the barbecue meat plant had really high phytoestrogens, probably the beans, but we're not exactly sure what was causing that. One of the dairies was really high. The other 2 had nothing. And we did end up figuring out what that was, and that was that they were also making soy-based supplements in addition to doing the milk. In general, dairies are not a problem. The soy products were high, as one would expect. And so this was pretty interesting. We also looked at a few cities where they got a lot of effluent from, like a big soy oil plant or biodiesel plant in town. And what we found was that some of the influent concentrations of these phytoestrogens were really high, 50,000, 18,000, 38,000, really high levels of these compounds. For the most part, they were degraded really effectively. But in one case, one of these cities was discharging phytoestrogens. and this is phytoestrogens only, we're not even looking at the estrone on top of it, that was above the level where we would expect to see fish impacts. And so even though they're doing a really good job, it's not good enough to get us down to those low levels that we need to be. So kind of the messages from this, we found that estrone was responsible for most of the activity, and that these food processing and soy-based industries can have really high phytoestrogen levels leaving the plants. And the take-home was that multiple sources are there. Sometimes they're not predictable, like this one dairy or the barbecue meat plant. But estrone does seem to be one compound that we can really focus on and try to accomplish some things there. So now I'll kind of move on and talk a little bit about understanding the fate and degradation of some of these compounds. And I'll take you back to that one slide. And what you can see is that sometimes degradation is pretty darn good, and sometimes it's not so good. It's really variable. And we see this nationally. Other people have seen this in the literature, that estrone in particularly sometimes degrades well, sometimes it doesn't degrade well. We saw the same thing with the phytoestrogens. And so the question is why? And so we really need to understand how and why these compounds biodegrade in the wastewater treatment plant, because that's the way that they're actually going away, is this biodegradation. So here's my complicated cartoon. So there are a couple of different models for how these estrogenic compounds could biodegrade. The first one is this little white bacteria, this little white Pac-Man, and this organism is a specialist. It only degrades estrone. That's it. That's all it eats. And if this is the case, we don't know which one of these is the case. If it's the specialist, then if there's not enough estrone around, they're not going to do very well, and they're going to pitter out. The second group of bacteria, the rainbow-colored bacteria, we think of as a multiple substrate utilizer. These bacteria eat estrone and the red dots and the blue dots and the light blue dots. They eat it all. But what we know about these types of multiple substrate utilizers is that they like food in low concentrations in general. So they'll eat lots of different things. They're like my daughter, but they want little amounts of everything. So they'll eat it all. The last bug is this light blue bacteria, and this organism eats the light blue dots. But they're like a golden retriever. They'll just, and sometimes they'll mess up and they'll eat an estrone molecule. So they're not very specific, but the thing they eat is the light blue dots. And so with these organisms, they need plenty of this other preferred substrate in order to survive and do well. The estrone's not going to help them at all, but we'll get lucky and they'll eat some of it if it's around. And depending on which one of those models it is, it's also important with respect to time because it's going to tell us what happens with respect to growth, which organisms grow, versus selection. Do we get preferential growth of one of these types of organisms over the others? And that's also going to be really important for how we might try to re-engineer the system. So we've run a ton of experiments in the laboratory, and this is my gratuitous reactor picture. I've got all sorts of reactors in the labs. We've used real wastewater, fake wastewater. We do clean water controls to make sure we know what's sticking and what's not. And we've done all sorts of things. And I'm going to talk you through one experiment because I think my student David did a beautiful job kind of thinking about how to run a clever experiment. And what he did was he had bacteria from wastewater that had not seen estrone. They were just growing on clean fake wastewater with no estrone in it. And then he had bugs that came from the same place that had been growing on estrone. So they had been seeing it and growing on it. And he did that for 30 days. And then he added estrone to all of them and watched it go away. And that's this data. And so the open symbols, if you can see them, it's a little hard to see. The ones kind of at the top, those are the ones where they were grown for 30 days with no estrone. Never saw it. And then the closed symbols are the ones where they were growing with it for 30 days and then they had more added. So the first thing we see is that these are non-specialist bacteria because there was degradation in all the reactors. And if it was our white bacteria, our specialists, they would not have been there in these open circle ones after 30 days. And so we know that these bacteria don't only eat estrone. So that's not the case. We also can figure out that there's actual metabolic degradation of estrone. That means they get energy from it. They get something from it. And the way we can figure that out is because there's this lag phase. If it was our golden retriever light blue bacteria, the second we add that estrone, they're going to gobble it up because it's nonspecific. They're not going to have to ramp up and make some new enzyme in order to degrade it. And so we see this lag phase before we get degradation, and that tells us that they have to start up new enzymes and get revved up in order to get energy from that estrone. So it's not our light blue bacteria. And then the last thing we see is that this ability to degrade estrone seems to improve their fitness. So the ones that have been growing on estrone for 30 days are better at it. They've gained some advantage. And so what we know then is that it's these multiple substrate degraders. They're getting energy from the estrone. They specifically have to degrade it with their own special enzymes, but they can degrade other stuff too. They can live on lots of different things. And David did a bunch more experiments that really showed this nicely. He also did a bunch more work that showed that in the short term, high concentrations of substrate don't do any harm, which is again consistent with those rainbow bacteria. They'll eat all the other colors, but they like low concentrations. So over the long term, if you give high concentrations of one of those other kinds of substrates, they're not going to do very well, and you'll get selection for a different kind of bacteria. But if we have low concentrations of food, lots of different things at low concentrations, then our estrone degraders are going to do really well, and they're going to thrive. We also found out in kind of some interesting experiments that he did that if you kind of cook the bacteria, you let them kind of sit there for a while, they produce waste products, and they die and they break open. All that stuff that they produce is really good at stimulating estrone degraders. So this kind of hard, yucky byproduct of bacteria works really well to stimulate them, which is kind of cool. So we know we've got our multiple substrate degraders. We get selection for the wrong bacteria if we feed them too much food. And that if you have these other bacteria producing carbon, that carbon is really great for our target bacteria. So take-home point here, now you're getting my whole it's complicated thing. Exposure matters, carbon matters, both the quantity and the quality. And differences are seen over the short and long term. But it's all consistent. It all makes sense. We've also done a ton of work on phytoestrogens. And what we saw was that it degraded really easily in both wastewater treatment plants and in model surface waters. But it degrades more slowly when it's cold, which is completely predictable. And so the take-home point with this is they're going to degrade easily. It's going to be slower when it's cold. So what can we do? All of this gives us some really good ideas of where to go. We can increase residence time. That means we can let the bacteria sit in there longer so that low carbon is present and we get good estrone degraders selected for. We can develop treatment systems that promote the presence of this microbially derived substrate, which is the lysis products and the waste products from our bacteria. There are new systems where we can kind of hold those bacteria in there for a long time and they produce a lot of this stuff. That would be a great thing to incorporate. We can provide pretreatment at industries that are potential dischargers of estrogens, like the barbecue meat plant. And we can extend treatment when it's cold. And so this gives us some really good ideas of things to do. Problem is that the estrogen degradation and presence is complicated. We can't guarantee success. This is really expensive. Holding those bacteria in there longer, adding oxygen, is expensive. The Metro plant spends about $1 million a month on electricity. It's expensive. These compounds aren't regulated. So, and I think that regulation might be very hard or impossible and probably not necessarily a good direction to go in. I think it might be a Pandora's box. So you're kind of suggesting that a plant might spend a lot of money to degrade something that's not regulated. They don't have to degrade it. And all this would be done to prevent fish feminization or masculinization, premature muscle spawning, protect tadpole development. So is it something we should try to do? So people always ask, and I'll jump to the question first, what do we see with humans? Because, we care about fish, but, they're fish, right? So we're seeing things in humans. There's been some really, really well done work that's shown that there's been a decrease in the age of puberty onset in girls. And this is reproducible, statistically significant, really solid data. Girls are going through puberty earlier. Sperm counts are going down. And this work was really controversial. It went back and forth. And the woman that did this study just kept getting more and more and more studies. And, it's human population. So the error bars are huge. But the statistics are solid. And there's a significant decrease in sperm count over time. Why? Is it diet? Is it stress? Is it chemicals in the environment? If so, what's the exposure pathway? Stuff we put on. I can guarantee you it's not wastewater effluent. These issues are not being seen in humans because of the stuff that I'm working on. The stuff I'm working on impacts fish, but it gets back to that question about trying to incentivize wastewater treatment plants to spend a bunch more money to degrade this stuff. It's not going to protect humans. So is there some inherent value in a protected ecosystem? We can't use the human card. Should we be doing this? And I think so. I wouldn't be doing this otherwise if I didn't think there was some good to come of it. And so based on this, what we've been doing is trying to take this slightly different perspective of, well, let's think about what might benefit a wastewater treatment plant. Spending a bunch more money to get rid of a compound that's not regulated is probably not going to be an easy sell. So if we're clever, we may be able to solve this problem while we're also solving other problems. And so that's where we've been working more recently. And there are a lot of drivers out there to change wastewater treatment. This is a fake check from 2010. This was the cost of electricity for wastewater treatment in the U.S. $2.8 billion for 1 year to treat wastewater in the U.S. We're spending as a country a huge amount of money to treat wastewater, the way we've been treating it 100 years, because that's what we've been doing. Also, we've got a lot of population growth in city centers. Our global population is urbanizing. The U.S. population is urbanizing. And so that also gives us an opportunity to have this big concentration of wastewater that maybe we can do something different with. And so where our current work is we've been focusing on low-energy wastewater treatment technologies, and we've been looking at lots of these low-energy treatment technologies, maybe treatment technologies that actually produce energy. And we've been looking at them with an eye to which ones actually work well with estrogens, too. And what we've found is this funny thing that looks like a little orange brain. This type of bacteria is called Anammox, anaerobic ammonia oxidizers, and they're bacteria that was discovered in the mid-90s. They had never been known of before. And this is some work we've been doing in the lab, my gross-looking reactor. And what's cool about this Animox system is it removes all the nitrogen, which then helps the Gulf of Mexico. It helps ocean productivity. It uses very little energy to do this. So we've got really good, this is our percent nitrogen removal. We're up between 80 and 100% generally. And then we looked at how estrone degrades in this system. And you can see there are these little black dots right on the 0 mark. So we're feeding 10, and pretty much nothing's coming out. So we're degrading our estrogen. We did some experiments. I showed you in the beginning, or I told you in the beginning, how women produce 3 different estrogens, and one of them turns into estrone. Well, under anaerobic conditions, which this is, estrone will turn back into that one. And so we looked for that. Are we making this problem just switching what compound we're making? And it's not there. And so it looks like we're completely getting rid of this stuff. We're starting to work again with my fish biologist colleague to expose fish to the effluent of this reactor to see, is there anything else in there we're missing? I've got a postdoc that's trying to dig into the analytical chemistry and figure out what it's turning into. But it doesn't look like it's turning into a problem, and it degrades really effectively. And so for us, we feel like we can take this perspective, this science and engineering perspective, and we can think about how we can solve the problem based on what we know, but make it easy for people to implement it and actually solve multiple problems at the same time. So it's really complicated, but we think that through science and engineering research, we can lower energy use, lower CO2 emissions, lower the cost of wastewater treatment, while also enabling ecosystem protection. And all of it, we had to work through all those complicated steps to figure out how it's degrading and why, to figure out, okay, we probably just need to do something different. And that's kind of where we're going right now. So with that, I'll acknowledge my collaborators. Heiko Schoenfuss is, he's a fish biologist that does a lot of really nice work on how compounds affect fish. Bill Arnold is a colleague in the Department of Civil, Environmental, and Geo Engineering that I've done a lot of the phytoestrogen work with. And then Deb Swackhammer worked on the early stuff with me, looking at what's coming in and out of wastewater treatment plants. My graduate students, Nate did all the phytoestrogen work. David did a lot of the estrone work that I showed you. And then Kira, who's now in Seattle, in beautiful mountains, did the work on the new technologies, the low energy technologies. And the funding was from the Environment and Natural Resources Trust Fund from the state of Minnesota, and then also our MinDrive program, which is the state of Minnesota as well. So with
I have
that,
a question.
yeah.
You probably explained, but I didn't understand. What's the difference between low energy treatment plant and the regular treatment plant?
Yeah, no, I didn't explain it, Betty. That's a really good question. So,
You want to repeat the question?
oh, sure, I will. Yeah, so the question was, what's the difference between a low-energy wastewater treatment system and a normal wastewater treatment system? Let's see if I can get back up to my... Sorry. I was trying not to do this to you, but... There we go. Okay, so... in a normal wastewater treatment system, what we do is in the secondary treatment, so we settle it, that's all normal, but then the secondary treatment, what we do is we blow a ton of oxygen into the system. And so that's where all this electricity cost is blowing air. It's just air, but we blow all that air in there so that our bacteria have something to breathe while they're eating our target pollutant. The way we're changing things with the lower energy wastewater treatment system is we're not giving them air to breathe. We're having them breathe something different. CO2, or in the case of these Animox bugs, they actually breathe nitrite. So we have them breathe different things, and so we don't have to supply the oxygen, so that cuts down on our electricity addition a lot. And then the other thing we can do is there are different ways of treating the wastewater that we can take this and make methane out of this part, or we can run the whole thing in a way that we make methane from the bacteria as well. That's one of their waste products. And so we can generate methane from the wastewater itself, or we can just work it in a different way so we don't have to add oxygen.
So now, how is the decision made if you do the regular treatment or the low energy treatment?
Yeah, it's a good question. So the question was, how do we make the decision whether we do low energy or the regular? If you've got a system like at the Metro plant, I don't know if any of you have ever been out there. It's a really
Very huge.
great place to go to. It's enormous. 280 million gallons of wastewater a day goes through that place. So they're probably not going to, they're, I think, very innovative. They're trying new things. They might put in a little pilot plant and see what they could do. But I think there'd have to be a pretty big upside for them to change because they've got so much infrastructure. But if you've got smaller plants, or smaller town, yeah, they could probably do some of this. It may be, we're working on some other things, trying to figure out how to make retrofits easier. And so there are a lot of people around the country working and trying to do new ways of anaerobic treatment where you could presumably just cover it and put in a kind of a membrane filter, and you could hopefully retrofit an existing tank and make it work. So I think
It's not the norm.
no.
Okay.
No. And there's also the potential, I think, to leapfrog what we've been doing for 100 years in developing countries, like they've done with the cell phone. They didn't go through the whole landline thing. And maybe we can leapfrog from very little or no wastewater treatment to really cool, low-energy wastewater treatment without even going through the middle stuff.
Your work was done in municipal treatment plants. Did those results translate to septic systems?
That's a good question. The question was, do these results transfer to septic systems? No. So septic systems generally are anaerobic, and they aren't doing the cool orange brain stuff that I was showing you at the end. And so the type of treatment that happens at wastewater or in septic systems is anaerobic, and you tend to not get much degradation, if any, of these sort of pharmaceuticals, estrogens, things like that. There are new add-ons for septic systems where you can add an aerobic chamber. I think they're probably pretty expensive. Yeah, and I was just actually talking with my colleague Heiko yesterday. He's done a lot of work on septic systems where they can actually see compounds coming up into lakes in certain areas, and so that is an area of concern, and that's something that I think we as a community need to start thinking about, the research community. How can we come up with easily implementable, cost-effective solutions for septic systems, because there are so many of them.
You know, in the land of 10,000 lakes, I would think money would be available.
Yeah. Yeah. Yeah.
Can you say something about the impact of these compounds on drinking water treatment systems?
Yeah, that's a great question. What's the impact of these compounds on drinking water treatment systems? They've found some of these compounds in drinking water treatment, or in drinking water, finished drinking water, treated drinking water. They've found there's a compound, carbamazepine, which is anti-epileptic, and that compound does not seem to degrade at all. There are people that are coming up with new ways of trying to get it to go away, but that one tends to be around. That one they've seen in drinking water. There's a flame retardant that they've seen in drinking water. They've seen estrone in drinking water. So some of these compounds they'll see in drinking water, and there's again really low levels. The thing I like to point out with the phytoestrogens, these levels that I had, let me see if I can get there quickly and not make you go blind while I scroll through. So, yeah, this compound, or this, so the highest we saw with the soy milk, that's the amount you'd get from 1 tablespoon of soy sauce. So this is not anything to freak out about for us. It's more because the fish are soaking in this stuff. It's problematic to the fish. And I would say some of the same things are true with respect to the drinking water. We don't know for sure. And I think if I were pregnant or I had small children going through puberty, I would be maybe more nervous. But I think the exposure route is really compounds we put on our skin, compounds we get from our couches, our mattress pads, household dust. That's where I think the exposure is much greater, not in drinking water. And I'll say, I live in St. Paul. St. Paul has activated carbon in their drinking water treatment plant, and that does a good job of removing these compounds. So I feel like there's the best water. I like the taste of it too. Not everyone does, but I think that St. Paul water is really, really, really high quality. And Minneapolis actually is looking at putting in the activated carbon too. They do it for taste and odor problems, for that kind of funky smell you get in the spring and summer. But it does a good job on these other compounds too. Yeah.
So how much soy sauce was that? You better go through that one more time.
If you had 1 tablespoon of soy sauce, it would be this level basically. And so,
Fish, not the human.
yeah, so that's for a human, but for a fish, that's much more problematic because they're living in it.
Just as a related question, the nitrate reduction that they had to put in the Des Moines water plant, is that using high energy, low energy? How does that work?
Oh, good question. So the question was about the Des Moines water treatment plant, and they have to remove nitrate because it's in the surface water that they're then treating for drinking water. And nitrate can be very problematic. That's, blue babies, it can cause your hemoglobin to not carry oxygen very well. And so I think they're doing an ion exchange, which would be very expensive. Nitrate is hard to remove. It's not very specifically removed. Other things that are charged are easier to remove than nitrate. So nitrate tends to be pretty hard to remove. So I don't know that it's very energy intensive, but it would be very expensive because they'll have to have these, I think they do ion exchange, these ion exchange towers, and they'll have to replace the resin and renew it. So it's an expensive proposition. And nitrate is a really hard one to deal with. Yeah.
Roundup, atrazine, and I just looked from a personal thing. We lived outside of Austin. There used to be a migration of frogs. Thousands upon thousands would migrate now. And same with worms. We don't have them anymore. We've got this Roundup and atrazine in drinking water. And I'm very concerned about it. What is your group doing?
So the question was about atrazine and Roundup and the impacts of that. Well, I went to grad school in Iowa, and it was before they had their new treatment plant, and we would always have atrazine in the drinking water in the spring. I think the atrazine's been incredibly controversial. There have been 2 scientific sides duking it out for years. I think the data is pretty compelling, and it makes me wonder why atrazine is still available. I know.
Monsanto and DuPont.
Yeah. So I think atrazine is problematic. I think they've shown pretty significant ecological impacts on frogs and other biota. And Roundup, there is some really interesting work going on right now with Roundup. I don't work with either atrazine or Roundup. I did my master's degree on atrazine and Allicor, but that's been a long time ago. But they're finding some interesting things with Roundup, and they're also finding, the last thing I heard, and this again is not something I work on, but that Roundup can exacerbate the eutrophication of water bodies, freshwater, inland water bodies, and cause more of these problematic algal blooms, which then cause those microcystins and these algal toxins, which shut down Toledo's water supply. And so I think that there are a lot of people looking at that and trying to figure out what role these compounds play. Yeah. Yeah, Denny.
I'm kind of curious, this looks like it'd be fairly expensive to reduce this for the result, other than if you're a fish or something like that. And then you mentioned like atrazine and Roundup and stuff. You know, the world population continues to grow, longevity continues to grow. It gets to be, you know, we always say they don't pay attention to science and engineering when they make decisions. But this gets down to really a political decision as to, are you going to feed the world, and you're getting longer life. So it's really a complicated thing to decide what's the right thing to do.
Yeah, it's very complicated, and that was my punchline, right? It's very complicated, and not just from the science. It's very complicated to make these decisions. I think some of the things we can think about with respect to wastewater is we should probably be doing anaerobic treatment. We should probably be trying to remove certain things from the wastewater that we can. We should be taking the brewery wastewater, the dairy wastewater, all of these high-strength wastes, and we should be treating them separately and generating energy. We shouldn't be dumping them into the metro plant. I think they're responsible for a large portion of that electricity use. And so if we can take care of things like that, I think that's pretty effective. There have been some in Europe, but then also in the U.S., they've been playing around with the idea of urine separation. A lot of these compounds are in urine. They're not in the fecal matter. In the fecal matter, you've got a lot of phosphorus. And so if you can separate the two, you can actually work, I mean, it's kind of gross to think about, but you can work with these things as a cleaner product that you can actually do something with. And so you could mine your urine for fertilizer. You can do a better job treating it for these kinds of compounds because you don't have all that high carbon in there. And so there are some really, I think, clever things we can do if we start just doing them. There's a wastewater, it's a group of plants down in Norfolk, Virginia. If any of you ever want to go see really cool wastewater treatment plants, Norfolk, Virginia is a place to go. They're doing everything innovative down there. They're doing this Animox with the orange brains. They're doing urine separation. They're doing injection into the groundwater and reclaiming that water. They're doing some really interesting things with their permits because they're discharging to the Chesapeake. I think they're recovering their phosphorus and selling it as fertilizer. Really, really cool stuff down there. And so, and there are some really big plants. We can do that. We just need to have, I think, the political will to get out in front of it and not wait until we're told to. So I think there are some solutions with wastewater. But the food thing, yeah, well, I think we have to use some of these, yeah, there's a balance. And there's some really neat work going on at the Institute on the Environment, looking at how to deal with marginal lands, where can we increase yield, how can we be smarter about where we crop globally. So hopefully, that's not what I do, but hopefully, really super smart people are moving the ball down the court there too.
If you're going to separate and collect urine, for example, separating and stuff, we can't even get money in this country to repair infrastructure. So it would be, in my mind, extremely difficult to start that complete separation.
It could be. I think, again, back to what Betty said at the beginning, we have this opportunity maybe in smaller communities or in the developing world. Why not?
Where you can maybe develop it before you try to use it full scale, on more of the population.
Yeah, exactly. Do we have time for one more?
I
Oh, I'm being cut off. Sorry. I'll stay. Yeah. She's cutting us off, but I'll stay and I'll talk with you. Thank you, guys.
Do I need